Hello, everyone. My name is Hayk Barseghyan. Welcome to the Optical Mapping Symposium. What I'll be talking to you today is about using optical genome mapping as a way of identifying pathogenic structural variants in several different types of genetic disorders. So first, I'd like to show you our workflow that we utilize at Children's National Medical Center. We start by, as everyone would, with either blood, cell, or tissue cultures. We use Bionanogenomics SP kit for high molecular weight DNA extraction. When we use the same DNA that we extracted for direct labeling and staining for Saphyr instrument, and we use the same DNA for PCR-free library preparation for short-read sequencing with Illumina NovaSeq and S4 flow cells to generate both FASTQ files that contain the short-read sequences as well as BNX files that contain molecule files for optical genome mapping. Both of these files are used to perform variant calling, and we use two parallel automated pathways to do that. One is utilizing access from Bionanogenomics, and the second using BaseSpace or Genox platform that utilize cloud computing such as AWS and Azure to perform short-read alignments and SNV and indel calling. Both of these pipelines generate two different files. One is SMAP containing structural variation information from optical genome mapping, and the second one is containing the short-read variants that SNVs and indels in the VCF format. We use these both files to perform integration, where we use the Genox platform that looks at structural variants identified by optical genome mapping and short-read realignment in those SV regions to provide better SV annotations. And we use Nanotator, which is an in-house developed R package to perform frequency calculations and additional annotations for structural variations, along with Bionano access, as well as we use Genox platform for SNV and indel annotations. And we basically, with this workflow, we're able to identify structural variations, copy number variations, single nucleotide variations, and small insertions deletions that may be disease causing. So I'd like to show you several examples of diagnoses that we were able to achieve with optical genome mapping. Here's case one, which is a disorder of sex development. This patient presented in clinic with learning difficulties and intellectual disability. He had low muscle tone, delayed growth, hypogonadism, and infertility. And the clinical team identified that the likely predicted diagnosis for this case would be Klinefelter syndrome, which has a karyotype of 47XXY, so presence of an extra X chromosome. So we performed optical genome mapping on this case, and what you see here is a summary of structural variants identified by optical genome mapping. On the left side are all the filtration criteria that we've used to filter the identified structural variants based on the quality scores, presence in reference databases in terms of frequency and size cutoffs. And what we see that these are all the different types of structural variations that we have identified in this genome that are neatly organized in the circus plot that has the G banding patterns for each chromosome from the karyotype. Then we have the structural variation track that is highlighted here for each chromosome, and then copy number variant track here on the third layer. And what we can see readily from the circus plot is that we can see that the X chromosome here has a gain of approximately 4x, and we can expand this look on the bottom here that shows the whole genome copy number variant view of all chromosomes, and we see that the chromosome X here has a copy number variant presence of 4 copies, and the Y chromosome here is present as 1 copy. And we've diagnosed that this case is 49XXXY, which is a more severe form of the Klinefelter syndrome. And it was a very quick and cool diagnosis. The second case is also disorder of sex development. This patient presented in clinic with Wilms tumor, an iridia, which is the iridium in the eyes, genitourinary abnormality, and intellectual disability. And the clinical team had predicted that this patient would have a diagnosis of 46XX Wager syndrome. We again run this patient on optical genome mapping platform, And on the summary page, you can see we've implemented the same filtration criteria as in case 1. And we see that this patient has a 46XX karyotype as evident by the copy number variant present in all of the chromosomes. And what we see readily is both in the circus plot and the copy number variant view of the whole genome, is that the chromosome 11, there is a copy number loss that is present in this patient. And what we can do is we can zoom in this particular location that has a deletion, and we can look at the actual genomic maps that were used, that were assembled for this sample. And what we see that the wild type allele for this patient is present here at the top, and the reference genome that is digested for chromosome 11 is in the middle. And we see that the second allele for this patient carries the 6 megabase deletion that was identified by de novo assembly as well as copy number variant analysis. And what we see is that this deleted region contains the number of genes that were important for this diagnosis, specifically WT1 and PAX6, that are responsible for genitourinary abnormalities, eye defects, and intellectual disability. Now, in this case, this case is 46XX. They will likely to have streaked gonads and that have fibrous tissue in them. And then last case for this sort of sex development is a patient presented in clinic with external genitalia of female-like, no palpable gonads, and no uterus by MRI scanning. Osteolytic bone disease, growth delay, elevated follicle-stimulating hormone levels, and luteinizing hormone levels. The predicted diagnosis for this case by the clinical team was that it's a syndromic form of 46XY gonadal dysgenesis, but there was no specific genetic disorder identified by clinical exome sequencing. So we performed optical mapping as well, and I'm not showing you the summary as with the other 2 cases. And what you see here is the chromosome 10 in the middle as a reference HT19. The gene that we've identified 6 kilobase per insertion is present in WDR11, which has been identified to cause hypogonadotrophic hypogonadism. Its inheritance pattern is autosomal dominant. However, we are still working with this case to identify where exactly the 6 megabase insertion is happening within WDR11. And the reason behind it is because, as we can see here with these labeled sites of the reference genome as well as the map assembled for this particular case, there's quite a bit of region where this 6 kilobase insertion could happen. And so we will perform a long-range sequence PCR in order to identify the location of this and see if it has any effect on WDR11 protein function. Okay, so I'd like to switch gears and talk about undiagnosed diseases. I have some examples that are very interesting. So first case is an 18-year-old male diagnosed with developmental delay, history of regression, short limbs, short stature, horizontal nystagmus, and myopia. Previous genetic testing was performed for this case, including chromosomal microarray that came back as negative, as well as genome sequencing that was also analyzed and came back as negative. We'll run this again on optical genome mapping, and what you see here is a summary of structural variants that are quite similar to other cases that I've shown. And what we see is that there's really no major abnormality that we can see with copy number variants. We see that this is a 46XY karyotype, and we've used the Nanotator, our in-house built tool, as well as Banana Access to do a further digging and see if there's any genes that overlap these structural variants that are shown in the circle plot here. And what we've identified is a 5 kilobase pair de novo deletion in the gene, in a CREB-BP gene. And what we can see here is that this is a heterozygous de novo deletion, where we see one of the maps of the proband having a clean alignment to the reference genome, showing that there is no structural variations. However, one of the alleles, you see that there is a shift in alignment that results in approximately 5 kilobase pair removal of DNA. And we look at the mother and father, and we see that both mom and dad, do not carry this deletion in their assembled alleles, both alleles. We see that with our analysis show that this frequency for this particular variant is 0 in reference databases, including database of genomic variations by non-control database, our internal cohort database that has approximately 80 individuals, and the Saphyr database. When we look at the gene function, we see that there are a number of these syndromes associated with mutations in this gene with the inheritance of autosomal dominant. Looking at the phenotypes present in these two syndromes, we see that the program has a lot of phenotypic overlap, and we think that this is the likely diagnosis. Further looking at the location of this deletion occurring within the gene, we see that this deletion is likely to affect exons 21 and 20 of the canonical splicing of this variant. And we see that loss of function variation in this gene has a PLI score of close to 0, which means that any mutations resulting in loss of function are not tolerable in this gene. Looking even closer, we see that there is a little bit of DNA present between the labels of optical genome mapping of approximately 3 kilobase pairs. So the deletion itself is 5 kilobase pairs, but the distance between two labels is 8 kb, which indicates that the actual breakpoint locations of this deletion can be either can have an error of approximately 3 kilobase pairs. So in order to so these two exons actually are residing in HAT, which is the histone acetyltransferase domain of this protein, and which is an integral part for protein function. So it is very important to know exactly the breakpoints for this deletion in order to identify whether it has any impact on protein function. So what we did, we looked at because we had access to genome sequencing, we've used two different structural variant calling platforms, DELI and MANTA, to see if any of these short read-based structural variant callers would be able to identify this gene. And we see that both of them were able to pick up this 5 kb deletion. And we look at the reads, we see that these are mate pairs. So the left one and the right one have an alignment distance between the two that is much larger than the insert size of short read sequencing, which is typically about 500 base pairs. And we see that they align much farther from one another. And we also see that the copy numbers indicating by the read depth here is also showing that the region is deleted. So this kind of helped us to find the breakpoints for PCR validation, and we've generated some primers for validation. We did program father and mother with PCRs, and we amplified approximately 1 kilobase pair in size band. We've done the Sanger sequencing, and we've seen that we're able to find the exact breakpoints for this deletion, which are highlighted here, chromosome 16. And what we see is that actually both exon 20 and exon 21 are deleted. This does not result in the shift of the frame of reading frame of this transcript, but we identified that the ClinVar has reported number of pathogenic variants in both of these exons. So we think that this is the likely diagnosis for this case. Second case of this is a 5-year-old male presented in clinic with epilepsy, starting at about approximately 1 month of age with multiple seizures, cortical visual impairment, severe hypotonia, severe developmental delay, where the child was not able to hold his head up, sit upright, and grab objects with his hand. The family history showed that the parents were unaffected, and the patient had an older brother that was also unaffected. Similar to case 1, this program had undergone chromosomal mark array, which came back as negative, as well as genome sequencing that also came back as negative. So again, we've performed optical mapping on this, and what we've identified is an approximately 90 kilobase pair deletion in CDKL5 gene. And this gene has been previously reported to cause epileptic encephalopathy as evident from a number of publications. But one thing that was looked a little odd was that optical mapping had identified this deletion that was present in 2 different maps on chromosome X, which is typically not the case because this case has a 46XY karyotype, and we typically see 1 map. But what we see here is we have the wild type allele, number of molecules supporting the assembly of that allele, and number of molecules that show the assembly of the abnormal allele. So what this really indicates is that the likelihood of this deletion to be present in a subset of cells carrying these molecules, so indicating a mosaic inheritance. We've looked at the parents, and both father and mother did not carry this deletion in their maps. As you can see here, the father has only 1 map that was assembled for the X chromosome for this particular region. And the mother has 2 alleles, so 1 allele here on the top and the second here on the bottom for these X chromosomes. So we wanted to make sure that the call was accurate and identify the mosaic level. So we've partnered with Mayo Clinic to do made pair sequencing, which confirmed the mosaic deletion that we see, approximately of 85 to 90 kilobase pairs in size, as evident with the circle here. These are 2 different reads aligning to 2 different locations. But in addition to this, it identified a mosaic inversion at approximately the same levels, 21% for deletion and 25% for inversion, as evident with the alignment of these blue and red read pairs. So what we did, we went back to this case in optical mapping, and we've started looking manually to see if there's evidence for inversion. And what we found is that the software was not able to align these 4 different labels into the location in the program map because of the deletion that was present here. But we can manually see that the distances for reference labels here and here on the program map 2 are aligning pretty well, indicating that yes, this is a complex rearrangement that is in mosaic state and is present in a de novo form, not present in mother or father. So here, what you see here are the molecules that are used to assemble this map2 that carries the deletion and an inversion. And we see that none of the parental molecules, including the maternal molecules and paternal molecules, are able to align, indicating the de novo origin. And there is total of approximately 23-24% of the molecules from the total number of molecules in the program that support this, indicating that approximately 24% of the cells are carrying this complex rearrangement. And so this yields to the diagnosis that this is the CDKL5 deficiency disorder, and many of the clinical features associated with this disorder are present in our case, including the epilepsy, severe global developmental delay, hypotonia, and intellectual disability. So the last case that I'd like to discuss is also an undiagnosed disease case. Many clinical symptoms and physical findings have been listed on the slide. Maybe craniofacial features, musculoskeletal features, blood and blood-borne forming tissues. So there's this case has many growth abnormalities. And so what we've identified by optical mapping is a inherited deletion from the mother of approximately 10 kilobase pairs in size in BMPER gene, that presumably takes out exon 9 and exon 8. But the inheritance for this particular disease is autosomal recessive, so we checked the short read sequencing data to see if there's any single nucleotide variants that could be present, could have been inherited from the father, or could have been have a de novo origin. And what we found that there is it's an exon 2 deletion. Single nucleotide variant that is de novo, and we looked at the classification, and it's likely that this variant is VUS. So it may have an effect on the splicing, but we're not sure at this moment. So it's this case is still work in progress in terms of the diagnosis. And when we look at this autosomal recessive BMPR gene, we see that a lot of the developmental growth abnormalities that were present in the case are also present in in patients who have been had mutations in PMPR gene. So with that, I'd like to end. Thank our collaborators from UCLA, Duke University, Disorders of Sex Development Translational Research Network, the Biological Genomics for support, and Genox for their support. And I'm looking forward to answering your questions in the Q&A session. Thank you.